Thank you very much. Yeah, really great first talks. I hope I can add something to this. So I'll be talking about the Brain Imaging for Global Health project, as Basilica said, and specifically our work in trying to understand neurodevelopment in rural Gambia. So while I'll be delivering the talk today, I'm doing so on behalf of a large number of people. Should be coming up on the slide. This one? Yeah. There we go. Yeah. So we have a huge team of researchers based both in the UK as well as in the Gambia, who've done all the work that I will be speaking about today. Of course, because of Basilica's direct connection to this project, we also have a really good turnout. Many of our UK team are here today. So well, I'll be giving you a little bit of a whistle-stop tour through some of our recent findings. If you're interested in any aspect in particular, do feel free to chat to any one of us, and we'll be very happy to talk further. So, of course, one of the goals of this line of research, other than wanting to understand neurodevelopment in a particular setting, is, of course, also to broaden the lens of developmental psychology as a discipline. So, of course, many of the things that we as developmental psychologists take as ground truths are actually based on quite a thin slice of the global population. I don't think I have to make that point to anyone in this room today. But I think because this work then often takes us into low and middle income settings, it's then often quite easy to exclusively focus on adverse experiences that can be encountered in these settings. However, in the talk today, I want to give a little bit of both sides of the story, really, and also talk a little bit about some of the different cultural influences that we might see that actually act as protective or resilience building factors, having to do, for example, with caregiving, family environment, and so on. However, what first sparked the interest in this project was that there are some poverty-associated factors in the Gambia that have been shown to affect child development across certain domains. One of the big ones being undernutrition. So you can see that illustrated here in these two pictures, where you can see the field station where all of our data was collected, and you can see that one of the pictures was taken in the wet season and one in the dry season. And coupled with the knowledge that many of the local population are subsistence farmers, it's then very easy to appreciate that there might be quite a stark variation in the availability of certain nutrients across the course of the year, which then thinking about pregnancy, infancy, and all of these key stages in development could, of course, play a role. And it has been shown that these factors do affect child development. So here we're looking at child growth, and you can see that rather than tracking on a straight line on group level, there is a frequent growth faltering over the first year of life. So we do see these effects on growth. And this then led one of our now PIs, Professor Sophie Moore, who's also here today, to ask the question, well, if we see these effects on growth, might we also see them with regard to brain and cognitive development? And the question for us then quickly became, well, if we're interested in brain development, can we actually use neuroimaging to study brain development in rural Gambia? So in a pilot that happened now 10 years ago in 2013, which I wasn't even involved in at the time, this was tested. So the group went out, took some neuroimaging equipment. I will be talking about what specifically was done a little bit later on, took it out to the Gambia. At the time, much of that journey was still over unmade roads, so we were all worried about the integrity of the system coming out at the other end. However, luckily, everything worked out really well. The lab could be set up in a matter of a couple of hours, and the first participant could come in and have their brain scanned. And also, broadly speaking, we found rather similar response patterns on the basis of this method as had been described in UK infants before by Dr. Sarah Lloyd Fox and Professor Claire Owen, who are also here. So this was, of course, really reassuring, and then led to the design of the much bigger brain imaging for the Bright project. You can see that we had cohorts in the Gambia of just over 200 and a smaller cohort in the UK of just over 60. And we had quite good coverage of sort of close proximity monitoring over the first 2 years of life. So we would start with some antenatal measures, but then also see the children in the lab to have their brain scans at various age points between 1 and 24 months of age. Thanks to Basilica, we then also were able to do a cross-sectional follow-up at 3 to 5 years of age called Bright Kids, which I will also be presenting some data of today. So of course, our focus was on imaging the brain we use two different methods for this. The first one being functionally infrared spectroscopy, which measures changes in blood flow in the brain. It has been pioneered for infant use, again, by Sarah and by Claire. And also we included electroencephalography, which was the main part of my own PhD research, which measures electrophysiological changes in the brain. Initially, we weren't interested in just understanding the brain. We wanted to understand the context of brain development as well. So we looked at a host of measures. I'll be pulling out little bits of data on some of these data. So for example, we looked at the home language environment. We looked at parental mental health, socioeconomic status, and things like that. We also had a large behavioral task battery, including eye tracking, an imitation task. We looked at parent-child interaction and also used a standardized behavioral assessment, which I think has been briefly mentioned before, the Mullen scales of early learning. If you're not familiar, it's very similar to the Bailey scales as well. And of course, for the aforementioned reasons, we also looked at nutrition and growth quite closely. So I think I made the point on a previous slide that we found that our equipment was robust and sort of portable enough to be used in the setting, but I also wanted to show this short video. I hope that's okay, Claire, because I think it also shows that the infants were also, quite happy to participate in these tasks. So you can see a 5-month-old here doing our EEG task. She's just sitting on mom's lap. It's all completely wireless, so very easy to implement, and she's playing with our very special research assistant on that day. So yeah, that seemed to be working out pretty well. Oh yeah, actually one thing that you probably couldn't hear because I kept talking was that with the EEG test, we presented very basic auditory stimuli, just a series of beeps essentially to see whether on the brain level we would see a response decrement to repeated stimuli and then a response recovery to novel stimuli. So this novelty detection was something we focused on quite heavily with both our EEG and our NIRS task initially. So this is from one of the first EEG paper that we published on these data. You can see here I focused sort of on the novelty detection aspect. So here we're looking at novelty detection between 1 and 5 months of age, because previous research suggests that children or infants become much better at novelty detection between 1 and 5 months of age. And while we did see that in the UK cohort, we didn't really see it in the Gambian cohort at the time. Since then, we've also looked at later age points, and we do see that they do show novelty detection at later age points. However, at these early stages, we saw sort of different, some slight differences with regard to these responses. Of course, we were also a bit, cautious. Maybe this was just something that happened on the EEG task. So we were very happy that we also had included an AfriBCD task looking at habituation and novelty detection. I won't go over the details of how it worked, but basically, infants were listening to a sentence of infant-directed speech that was repeated over and over before then being spoken by a new speaker. So again, we were interested to see whether there would be neural response decrements and then a response to the novelty. And you can see here in the top corner what the UK responses were. So we did see response decrements and a response recovery to the novelty, and we saw it from as early as 5 months of age. Whereas in the Gambia, we saw a slightly different pattern, which included sort of continued habituation to the spoken sentence, and it was regardless of the stimulus condition. So we didn't really see, an increased response for the novel neural stimuli. We did start to see that emerging at 18 months, which then led us think maybe there's a bit of a timing difference in when this neural specialization with regard to picking out novelty in this kind of stimuli occurs. So having spoken a little bit what we saw sort of purely on the brain level, I also want to talk a little bit about some findings linking early brain responses with later behavior. So this is from a paper published last year where we looked in both the UK and in the Gambia whether early habituation and novelty detection were associated with imitation at 1 year of age. And we found that while the overall levels as to how much novelty detection and habituation we saw across the sites differed, we still saw this association with later imitation in both sites. So that mechanism still seemed to be there. And of course, this is only looking at EEG at 5 months up to a year of age, so it's a couple of months of sort of this longitudinal association, but we also have since started to look at some of our preschool data, especially we focus on executive functions here. And we could see that from as early as 5 months of age, but also at 18 months of age, we could pull out things from the EEG and the NIRS data that were associated with executive functions at preschool age. So we do see these longer term associations as well. And also just to highlight some work by my colleague Chiara Bugarelli, who's not here today. She looked at functional connectivity, and she found that, I'm just going to summarize this because I think I'm going over time, but the degree to which there was functional connectivity between the frontal lobes was associated, again, with executive functions at 3 to 5 years of age. So I think this all shows that we can pull out really potentially useful information from all these neural measures that can then tell us something about how this brain will do later on. So I also said that I talk a little bit more about the family context. So this is from a paper using submitted manuscript that was led by Marlene and myself. And we were looking at LINA recordings. So if you're not familiar, they are small language recorders picking up how many vocalizations are emitted by the child, but also how much talk is around them. And another really useful measure that it gives you is the amount of turn taking there is between the child and others around him. And we could basically see that the degree to which there was turn taking at earlier age points was associated with subsequent child vocalization. So this early turn taking then drove later child vocalizations. So this made us interested to understand, well, what really drives, like what in the family, like what family, factors might affect how much turn taking there is in the first place. So one thing that we did was we looked at the number of adult caregivers that were present, that were with the child during the recording day. And we found that not so much at 12 months, but definitely at 18 and at 24 months, we would find this U-shaped relationship whereby there was the most turn taking when there was sort of a mid-level of caregivers present. So not necessarily when children were just with 1 or 2 adults, and also not when they were with too many, but if there was this mid-level, we would find that this led to the greatest number of turn taking throughout the day. And one other thing that I wanted to highlight is work led by my colleague Sam McCann and her supervisor Sophie Moore in exploring the context of undernutrition in this context, which is of course something that we were quite interested in from the start. So Sam looked at the Mullen scales of early learning, so our standardized behavioral assessment, and she found that there was a small but persistent difference, a 1.9 point difference between children who had the lowest versus the highest iron status. And this difference seemed to hold from as early as 5 months up to 5 years of age. So a small but sort of persistent change there. And then the other thing looking at the impact of iron was to do with our eye tracking data. So again, Sam looked at those with the highest versus the lowest iron status, and found that there was a difference in the reaction time, essentially, with which children engaged and disengaged with different visual stimuli, which was present during the earlier months and years, but then seemed to diminish into the preschool period. So these really interesting first findings on the impact of iron status then were in part what sparked a new project, again led by Sophie Moore. And if you're interested, definitely go talk to her and Sam, which is called the Indigo trial, which is a randomized control trial of nutrition supplementation, which is looking at whether this kind of supplementation has any effect on neurodevelopment and which kind of supplementation might be the most beneficial. So with this, I think I've just barely managed to keep to time. So I thank you, and if you have any questions, I'd be very happy to answer that.